Welcome back. I'm here to answer a question. And the question is, why males are more prone to X-linked disorders? In a better way, I can put it in a better way. Why males are more prone to X-linked disorders whose genes are located on differential region of X chromosome. The question is, why males are more prone to X-linked disorders? Why males are more susceptible to X-linked disorders? And I am putting it in a much better way. Why the question should be, why males are more prone to X-linked disorders whose genes are located on differential region of X chromosome. Before I will answer this question, you must understand some basic facts and basic things which are very important in genetics. I will tell you, for example, I will take any chromosome. Let me take chromosome 2. Chromosome number 2, for example. Chromosome number 2. Since when we discuss human beings have 23 pairs of chromosomes. 23 pairs of chromosomes. And when I am just taking out what chromosome number 2, I am just taking out chromosome number 2, and chromosome number 2 must be a pair. It must be a pair. And the pair of chromosomes are known as homologous. They are exactly identical. Centromere is at one place and the centromere corresponds to each other. And the chromatids are also identical in size and shape. So if I will take any pair of chromosome, as I have taken the chromosome number 2, the two I can say homologous chromosomes. The two homologous chromosomes of this pair are completely identical. Right? If I will take chromosome number 3, maybe chromosome number 3 may like this. It may be like this. The chromosome number 3 is a pair. It is a pair. And the pair is completely identical with the pair is completely identical to each other. But if we just see the morphology of chromosome number 3 and chromosome number 2, it is different. So I can say any homologous chromosome pair is completely identical in structure. Therefore, when I discuss chromosome number 2, it is a pair and the pair is completely identical. Now, we also know on these chromosomes we have genes. For example, if we have here a gene, we have also a gene here. For example, if we have another gene here, we have another gene here. On the chromosomes, you will find genes. And the genes have alleles. If we see the Mendelian laws of inheritance, Mendel said that for any trait, for any character, there is a gene. According to Mendel, if we just see any trait, for any trait there is a gene, and for any gene there are two alleles. This is allele A1, this is allele A2. So for the expression of any gene, there should be two alleles, and these two alleles will be determining a trait. The two alleles will be determining a trait. For example, according to Mendel, if we say plant height, plant height is a trait. Height is a trait. Height is a trait. And this trait is controlled by a gene. And this trait is controlled by a gene. And this gene has two alleles. And the two alleles may be T or T. The two alleles may be capital T and small t. The two alleles may be small t and small t. See, just I am telling you the basics. If we have a trait, and the trait here I am discussing is the plant height. Plant height is the trait, and this trait is governed by a gene. It has a gene, and this gene has two alleles. The two alleles may be either TT, the two alleles may be capital T and small t, or the two alleles may be small t or small t. The expression of these two alleles is, what is the expression of these two alleles? If these two alleles are present, the plant is tall. If capital T and small t is present, the plant is again tall. And if small t and small t is present, the plant is dwarf. Basically, when we want to determine the plant height maybe the plant may be tall, plant may be dwarf. It will depend upon the presence of the two alleles which are basically governing a gene, which are basically expressing a trait. If any plant has capital T and capital T, it is a tall. If any plant has small t and small t, it is a dwarf. And if any plant has capital T and small t, it is again a tall. Then we call this condition, capital T and capital T condition, as homozygous. As homozygous. We call this condition, small t and small t, also as homozygous. Homozygous. Whether it is capital T and capital T, when both the two alleles are identical, we call it as a homozygous condition. So this is homozygous, this is homozygous. But it is homozygous dominant. Homozygous dominant because it is expression here in case of capital T and small t is tall. I will just tell you. It is homozygous dominant and this is homozygous recessive. It is homozygous recessive. Let us see this. Here we have capital T as well as small t. But the capital T is a dominant allele. The capital T is the dominant allele. And this allele, when comes in combination with a recessive allele like small t, this capital T is going to dominate the small t, and the overall expression of the gene, overall expression of the alleles will be exhibited by this dominant one. So we can say it is dominant, or we can say it is heterozygous condition. In the heterozygous condition, the expression of trait will be exhibited by the dominant trait. If we have capital T and small t, the expression of small t will not come. However, the expression of capital T is definitely going to get noticed. So this is the basic thing which I need to discuss, because I have to say you something. Similarly, let's come back from this to this. We'll come back to this. I told you we have chromosome number 2, and this chromosome number 2 has the alleles, has the genes, and this is a homologous pair. Suppose we have a gene here. This gene may be controlling some character. We have another gene here. It may be also controlling some other character. Another gene here. It may also be controlling some other character, and so on. When we see the gene has two alleles. Just I will put this kind of example I will put here. For example, if it is capital T on this side, on this chromosome, the allele is capital T, and on this chromosome the allele is again capital T. Therefore, for looking to the character, what type of character they are exhibiting, we have to put our eyes on both the alleles. We have to see basically the genotype. I will see it is capital T. What I am observing, it is capital T and it is capital T. Yes, we know the plant height in this case will be tall. For example, if it is capital T, if it is small t and small t. For example, if it is small t, the small t and small t, they are in the homozygous condition, and the expression of small t and small t is in the form of height, the plant height, which will be dwarf. A dwarf plant will be born. Or if it is capital T and small t, it is heterozygous condition, and this will serve as a recessive allele, and this will serve as a dominant allele. Although this is a heterozygous condition, but the expression of character will be determined by the dominant trait, dominant allele. The expression of character will be determined by the dominant allele, and the dominant allele here is T. So again, the expression of this type of gene is again tall. We have a tall plant. So this is the framework which I am making you to make you understand why males are more prone to X-linked disorders. When we talk of females, let's end up this way. When we talk of females, for example, now let's discuss the females. In case of females, the sex chromosomes are 2. They have one X, X, and X. The female has 2 X chromosomes. It means if any gene is present here, if we will see some genes maybe on the chromosomes. For example, here we have a gene, here we have a gene, here we have a gene, here we have a gene. And if I have to predict the expression of any gene, I have to see to both of the alleles. For example, this gene is coding for something. For example, it is coding for normal color. The gene is basically responsible for color perception. The gene is basically responsible for color differentiation. Therefore, I have to look for this allele as well as this allele. I will see if this allele is normal and this allele is normal. We say the person must possess the perception of color. Therefore, the inheritance pattern of XX is exactly identical with the inheritance pattern of all the autosomes, because autosomes also have homologous pair. If I talk of chromosome number 1, it has a homologous pair. If I want to see what will be the expression of gene, I have to look on this chromosome also, on this chromosome also. And similarly, if I will talk of chromosome number 15 is a homologous pair, and for just predicting the expression of any gene, I have to look on this chromosome as well as on this chromosome. Similarly, just like autosomes, similarly, the X chromosomes, if we have a female, it has 2 X chromosomes, and for the expression of any gene, I need to see the allele present on this side and the allele present on the other side. That means on the homologous pair. Now, what is happening in males? Therefore, we can just conclude all the autosomal characters, all the autosomal genes, and the inheritance pattern of the XX chromosomes is exactly same. There is no difference. However, when we want to see the males, let's see male. Male has XY chromosomes. XY chromosomes. For example, this is X chromosome. X chromosome is very long, containing large number of genes, and we have Y chromosome. Y chromosome is small. Y chromosome is small in this way. This is a male having X chromosome and having Y chromosome. This is not the homologous pair. It is not just like X and X, which was exhibiting the homology. Here, what we are witnessing, what we are seeing, we have X and we have Y. There is a great degree of dissimilarity between the two. Therefore, we can predict the XY chromosomes have two types of regions. One is the homologous region, and second is the differential region. In case of a male, the XY chromosomes are not completely identical. It will exhibit, it may have some portion which is homologous, and it may have some portion which is differential, quite different. See, for example, if I would like to see this part of X chromosome, right, and this part of Y chromosome. This is X chromosome, and this is the similar part. This part of X chromosome is here, and we are just watching a similar pattern of structure on the Y. We can say this part of X and Y is homologous. This part of X and Y is homologous. Or we can say this is the homologous region of X and Y. And on the contrary, if we see this part, this region, this region of X chromosome is there, of course, but this region, there is no corresponding region, no corresponding pair, no corresponding homology on the Y chromosome. The region is only present, this region is only present on the X chromosome, and there is no homologous region on the Y chromosome. Therefore, we call it as differential region. Differential region on X chromosome. And one more thing, we are also looking here, we have a small part on the Y chromosome. We have the small part on Y chromosome, and this Y chromosome do not have a homologous part on X chromosome. Therefore, we will call this region also as differential region of Y chromosome. X and Y, in case of X and Y, we are looking for two types of regions. One is the homologous and second is the differential. Homologous region is this one. This is the homologous region. It is present on both X as well as on Y. We have the differential region which is present only on X chromosome, and we call it as differential region on X chromosome. And we have a differential region which is only present in Y chromosome, and we call it as differential region on Y chromosome. Therefore, genes will be present on this region also. This is also differential region. This is also differential region of X chromosome. Genes will be present on the homologous region, and genes also will be present on the differential region. The genes which are present on the homologous region, are known as XY linked genes. XY linked genes, because we must be having one allele here and one allele on the X chromosome and one allele on the Y chromosome. We call such genes as XY linked genes because the alleles are present both on the X as well as on the Y. So we have XY linked genes. And here if we talk of these genes, suppose here also we may be having genes, and these genes are only present, on the differential region of X chromosome, and such genes do not have the homologous gene. Such genes do not have a homologous gene on Y chromosome. Therefore, they are completely present on X chromosome, and we call such genes as X-linked genes. And then we have some genes. We'll be having genes here also, differential region of Y chromosome. and these genes do not have a homologous gene, they do not have a similar loci, they do not have a similar homology on the X chromosome, but the genes are completely located on Y chromosome, and we call them as Y-linked genes. Y-linked genes. Therefore, we have three types of genes. One is the XY linked genes, second is the Y-linked genes, and third is the X-linked genes. X-linked genes. We have three types of genes: X-linked genes, Y-linked genes, and XY-linked genes. If we see what will happen if we see, we will just check the inheritance pattern. What will be the inheritance pattern of XY-linked genes? For example, if there is any gene here, and it must have on the same loci in Y chromosome, it must have a corresponding gene there or corresponding allele there. And if I want to see the expression of this gene, if I have to see the expression of this gene, I have to look back on the loci, both on the X as well as on the Y. There's a trait, there's a character, and this character will be governed by the presence of, by the expression of, or by the presence of the two loci of the same loci which are representing a particular gene. Just like autosomes, as I discussed. For example, if this is capital T and capital T, if it is capital T and capital T, I can say the character will be tall. If it is small t and small t, I can say the character is dwarf. If it is capital T and small t, I can say this is heterozygous condition, and again it is tall, because it is dominant allele here and it is recessive allele here. Therefore, the expression of any trait, for the expression of any trait, I have to look to the both alleles, what is going on. And we have to see, suppose if this is capital T and this is small t, we have to see the capital T and small t. This small t is 50%. It constitutes 50%. This constitutes 50%. And this is the character which represents dwarfness. This is the character which represents tallness. But dwarf is not expressed at all. It is the tallness which is being expressed. Therefore, the expression of the small t is masked by the presence of capital T. Although the genetic contribution of dwarfism, the genetic contribution of this small t is 50%, but its expression is 0. Therefore, I can say maybe if we for any character, we have to go back to the homologous region. We have to go back to the homologous region, and we have to ascertain the feature, the nature of the alleles. However, when I talk of these X-linked genes will be transmitted, will be expressed. Suppose this is a male, this has this region, X-linked genes. X-linked genes are those genes which are present on the differential region of X chromosome. All these genes will be expressed both in dominant form as well as in recessive form. They will be expressed in dominant form as well as in recessive form. If we see this part, we have capital T and small t. If small t is there, small t is not going to express. But if this small t is present here, for example, the small t allele is here. For example, if the small t allele comes on this region, it is going to express in spite of its recessive nature. So we can say whatever genes will be present here, there is no opposition gene on the opposite side. There is no homologous part on the opposite side. In whatever form we will witness the genes here, in whatsoever form we will be having genes on this side, they are going to express whether they are dominant or recessive. Therefore, this is one thing. Second thing is, we have Y-linked genes. Whatever genes will be present on this region of Y chromosome, on the differential region of Y chromosome, whatever genes are present on this Y chromosome, on the differential region of Y chromosome, they are going to get expressed whether they are in dominant form or recessive form. If they are in recessive form, they will be expressed. If they are in dominant form, they are again expressed. Therefore, let's go back to our question. Why males are more prone to X-linked disorders? Or I told you, why males are more prone to X-linked disorders whose genes are located on differential region of X chromosome. If there is any defect in this gene, for example, we have color blindness, we have night blindness, we have hemophilia, we have Duchenne muscular dystrophy, we have Hunter syndrome. There are so many defects, X-linked disorders. We can say if we have any disorder whose gene is located, whose gene is getting defected and the gene is located on the X chromosome. For example, if we have color blindness, or we have hemophilia. Let's take hemophilia. We have XH and XH. It is hemophilia. The XX means it is a female and the female is hemophilic. The female is hemophilic. It means, for example, if this is a female, it must have X and X. The female must have X and X. And the hemophilia gene is located on X chromosome. Here it is hemophilic gene. Here it is hemophilic gene. If this female has a hemophilic allele here as well as a hemophilic allele here, then this female is going to be hemophilic. So we have to look for both the alleles. Again, we have H and it has a corresponding allele on the homologous pair. It is H. So here we have hemophilic allele here, hemophilic allele here. This female is hemophilic. But if this H is not there, for example, it is a normal X. On this side we have a normal gene, but on the homologous part we have a hemophilic gene. What will be the character? What will be the expression of this kind of trait? The female is not hemophilic, but it is a normal. It is a normal female. It is not hemophilic because hemophilic gene has arrived on one of the X chromosome. But on the second X chromosome, there is a normal gene, and this normal gene is a dominant gene, and the expression of this normal gene and hemophilic gene will be in such a way that the normal gene will dominate over the hemophilic gene, and therefore this female will be a carrier. But there will be no question of hemophilia. She will be born a normal female. She will be a normal female. She will not be hemophilic. But if at all this hemophilic gene comes here on the Y chromosome, if at all the hemophilic gene comes on the X chromosome of male, if at all the hemophilic gene comes on the differential region of X chromosome of male, it is going to express in all the cases. Therefore, females have tendency. Females have two X chromosomes. Females have two X chromosomes, and because of the two X chromosomes, if any recessive gene will come on one X chromosome, it will be dominated by the normal gene. But however, in case of males, there is no choice. There is no corresponding homology of X chromosome. Therefore, if the gene comes at all on the differential region of X chromosome, it is going to express. And this is the answer of our question: why males are more prone to X-linked disorders. But I just elaborated in this way that why males are more prone to X-linked disorders whose genes are located on the differential region of X chromosome. Here we will see this is the differential region of X chromosome, and the differential region of X chromosome in case of males do not have a homologous part. Therefore, if any defective gene comes, if any defective gene appears, the defective gene is going to express, although the defective gene was recessive, but still it is going to express because it do not have any homologous part which is going to mask it, which is going to dominate it. So that's why we can conclude that males are very prone to X-linked disorders, but not completely X-linked disorders. For example, when we talk of this part, X and Y linked disorder, the inheritance pattern of X and Y will be same as that of autosomes. Therefore, if we will see the gene, if any gene is defective here, so if any gene is defective, for example, we have a defective gene here. This defective gene if present on X and Y, or if present on X and X, does not have any difference. It does not have any difference because there is a homologous region, and in X and X also there is a homologous region. But when we talk of differential region of Y chromosome, differential region on X chromosome, differential region of X chromosome is present only in case of males. Therefore, it do not have a homology. And whatever genes get defected on this region, they are expressed. They are expressed in all forms, whether they are dominant or recessive. Therefore, males are more prone to X-linked disorders, but those X-linked disorders whose genes are located on the differential region of X chromosome. One more thing I will tell you, Y-linked genes. The inheritance of Y-linked genes is known as holoandric inheritance. Holoandric inheritance. Holoandric inheritance. Holoandric inheritance. See, we know it very well that at the time of gametogenesis, the homologous chromosomes pair up, and they exchange the segments by way of crossing over. Therefore, every gene is transmitted to the offspring from the parent, from the mother, but first they will involve the genetic recombination, and these genetic recombinations are responsible for the variations. But there is a small amount of genes, very small amount of genes. There are very few genes which are transmitted from father to the son without any genetic recombination. What are these genes? I am telling you, there is a small portion. There is a small portion. There is a small number of genes which are present in the father, and they are transmitted to the son. They are transmitted to the son without any genetic recombination. And such genes are known as Y-linked genes, holoandric genes. Holoandric genes are located on the differential region of Y chromosome. Because in this, suppose if we have a male XY, this male is going to produce the gamete, and we are looking for the gamete which contains Y chromosome. What will happen? At the time of gametogenesis, this homologous region of X and homologous region of Y are going to synapse. There will be crossing over, there will be genetic recombination, and definitely the gamete which will be produced will be having the chromosome with a difference. But when we talk of this region, the male possessing this region, the differential region on Y chromosome, the male which has this differential region on Y chromosome, the genes which are located on this differential region of Y chromosomes are not going to pair up because they have no homology. They have no homology here, and they are not going to pair up. Therefore, they will be transmitted directly from father to the child, better to say to the male son, without any genetic recombination. I hope the answer of the question is being delivered by me. I hope you should understand. Thank you very much.